Welcome to this lecture on biochemistry. Today, we are going to explore one of the most elegant and crucial mechanisms in all of biology, electron carriers. If you have ever wondered how the sandwich you ate for lunch actually powers your heartbeat and your brain without causing you to spontaneously combust, you are in the right place. We will build this understanding from the ground up, starting with a simple analogy. Now that we have introduced our topic, let us start with a mental picture. Imagine you are trying to transport a massive, glowing hot coal from a roaring bonfire into a small stove inside your house. You certainly cannot carry it in your bare hands because it would severely burn you. And you cannot just throw it because it might set the entire house on fire. Instead, you would use a series of specialized metal tongs and insulated buckets to carefully pass that hot coal from one point to the next until it safely reaches the stove. In your body, the hot coals are the high-energy electrons extracted from the food you eat every day. Building on our hot coal analogy, we can now formally define what these cellular tongs and buckets actually are. In biochemistry, electron carriers are specialized organic molecules or protein complexes designed for one specific job: safely transporting energetic electrons. They do this by reversibly accepting and donating electrons during metabolic redox reactions. When a carrier accepts an electron, we say it becomes reduced. When it gives that electron away to another molecule further down the line, it becomes oxidized. This constant cycling between reduced and oxidized states is what allows them to pick up electrons from your broken down meals and deliver them to the cellular power plants. Knowing how they function, let us meet the most common electron carriers you will encounter in cellular respiration. First is nicotinamide adenine dinucleotide, which constantly cycles between its oxidized form, NAD+, and its reduced form, NADH. Another major player is flavin adenine dinucleotide, which cycles between FAD and FADH2. You can think of these specific molecules as the mobile delivery trucks of the cell. They physically move through the cellular fluid, picking up high-energy electrons from the breakdown of glucose and shuttling them over to the electron transport chain, where their energy can be extracted. While NADH and FADH2 are mobile in the fluid, other carriers are restricted to the membrane. For instance, we have lipid-soluble carriers like ubiquinone, which is often called coenzyme Q. Because it is lipid-soluble, it moves freely within the hydrophobic or water-fearing core of the mitochondrial membrane. Finally, there are protein-bound carriers known as cytochromes. These are large complexes embedded in the membrane that contain iron atoms. It is actually these iron atoms that physically hold on to the electrons as they are passed down the electron transport chain, much like a relay runner holding a baton. You might be wondering, why do we even need these microscopic delivery trucks in our cells? Why not just react the food directly with oxygen? Well, if the glucose from our food were simply reacted with oxygen all at once, the reaction would be a literal biological explosion. It would release all of its energy instantly as unusable, dangerous heat, which would destroy the cell. Electron carriers prevent this dangerous explosion. By capturing the high-energy electrons and passing them down a chain, they ensure that the energy is released in tiny, highly manageable steps rather than one massive burst. Because the energy is released in these tiny steps, the cell can actually do useful work with it. At each step along the electron transport chain, a small amount of energy is safely released. The cell captures this energy and uses it to pump protons across the mitochondrial membrane. This creates a pressurized gradient of protons, which eventually flows back across the membrane to power the synthesis of ATP. ATP is the universal energy currency of the cell. Without electron carriers breaking the energy release into steps, complex life as we know it would instantly cease to exist. To understand exactly how much energy is released at each step, we first need to look at the electrical driving force, known as the change in reduction potential. Conceptually, this is like calculating the total height of a waterfall before figuring out how much power the falling water can generate. The formula is delta E standard prime equals the E standard prime of the acceptor minus the E standard prime of the donor. Delta E represents the change in standard reduction potential, measured in volts. The acceptor is the molecule receiving the electron, and the donor is the molecule giving it away. When the acceptor has a much higher affinity for electrons than the donor, this voltage difference increases, creating a stronger electrical pressure driving the metabolic machinery forward. Once we know the electrical pressure, we can calculate the actual chemical energy available to the cell using this fundamental formula. It connects the electrical energy of the moving electrons to the chemical energy used to do work. The formula is delta G standard prime equals negative N times F times delta E standard prime. Delta G represents the standard free energy change, measured in joules per mole. The variable N represents the exact number of electrons being transferred, which is usually 1 or 2. F stands for Faraday's constant, which is approximately 96,485 joules per mole per mole. When the voltage difference increases, the resulting free energy becomes more negative. A negative value means a larger amount of energy is released and available to make ATP. Before we move on, let us see this with a concrete example. We want to calculate the total energy released when 2 electrons are carried by NADH and delivered to oxygen. We are given the reduction potential of NADH as negative 0.32 volts and oxygen as positive 0.82 volts. First, we find the voltage difference by subtracting the donor from the acceptor, giving us 1.14 volts. Next, we plug this into our free energy equation. We use 2 for N because NADH carries 2 electrons, and multiply by Faraday's constant and our voltage. The result is approximately negative 220 kilojoules per mole. The negative sign indicates a massive release of energy, which the cell harnesses to keep you alive. Now that we have calculated the energy for NADH, let us look at a different carrier located much further down the respiratory chain, cytochrome C. We are given its reduction potential as positive 0.25 volts. Using the exact same logical strategy, we first find the voltage difference by subtracting 0.25 from oxygen's 0.82 volts. This gives us 0.57 volts, which is exactly half the voltage drop we saw with NADH. We plug this smaller voltage into our free energy equation, again using 2 electrons. The final answer is approximately negative 110 kilojoules per mole. Because cytochrome C holds onto electrons more tightly, passing them to oxygen releases significantly less energy, perfectly illustrating how energy is released in smaller, controlled steps. Having calculated the energy at different steps, let us now visualize this entire process. What you're looking at is a detailed schematic of the inner mitochondrial membrane. Notice the large protein structures labeled complex 1 through 4 embedded in the membrane. You can see the path of electrons, represented by arrows, starting from NADH on the left and flowing through the mobile carriers, like coenzyme Q and cytochrome C. Observe how at complex 1, 3, and 4, there are upward arrows, indicating protons being pumped across the membrane. This is the physical manifestation of the energy we just calculated. Finally, on the far right, you see the ATP synthase complex, which uses that built-up proton gradient to synthesize ATP. This diagram perfectly captures the beautifully orchestrated bucket brigade that sustains life. To wrap up our lecture, let us review the most important ideas we have covered today. First, electron carriers are the vital transport vehicles of cellular metabolism, acting like tongs and buckets to safely capture high-energy electrons from the food we eat. Second, by passing these electrons along a series of carriers with increasing affinity for electrons, the cell can harvest energy in small, safe increments rather than one dangerous explosion. Finally, this beautifully orchestrated bucket brigade prevents our cells from overheating and provides the exact amount of power needed to synthesize ATP. You have now mastered the fundamental biochemistry of how your body powers itself. Great job, and keep up the excellent work.